My name is Pam Rice. I'm very honored to introduce this year's BA Research Day as your new Deputy ACOS for Research. Thank you. I want to first thank the organizers of this year's event, Dr. Becky Scalzo, please. And also Dr. Amy Keller, who happens to be presenting her research in Sweden right now. So she can't be here to join us. They're both excellent representatives of career development awardees here at the VA. And I'd like to add that we're actually we have one of the largest numbers of career development awards of any VA research program in the nation. We're quite proud of that. And it's also a testament to the strong mentorship that we have available here at the VA. I also want to extend our thanks to Dr. Swati Patel, who helps co-organize this event and was instrumental in helping set up the Dennis J. Memorial Lectureship. Thank you so much for working with the CU and the Denver Research Institute to bring us Dr. Macrae as our lecturer this year. Dr. Patel represents, I would say, the cream of the crop of Dr. Ahnen's mentees, and a great example of the future of research. She is continuing his mission to help bring awareness of colorectal cancer screening and reduce the incidence of colorectal cancer deaths in our veterans. I am also one of the privileged to have been mentored by Dennis Ahnen as well. This is a little emotional, but I started my postdoctoral fellowship here at the VA in 1998 with Dennis in his lab. And it was during the 1990s, after I graduated with my PhD, that the NIH was really emphasizing the bench to bedside research. But it wasn't until I met Dennis and I started my work here at the VA that I realized this was probably the best healthcare system to actually implement bench to bedside research, and I still believe that today. I was really impressed, first of all, by Dennis's deep understanding of basic cell and molecular mechanisms and the genetics of colorectal cancer predisposition and progression. But it wasn't until later in my career with Dennis that I realized the breadth of his research and that it spanned his clinical practice, and even reached global proportions in his efforts to increase awareness of colorectal cancer screening, which is still the most effective way to prevent colorectal cancer. So from a bench to bedside research perspective, Dennis was the real deal. He was the whole enchilada smothered in green chili. So I wanted to highlight just a couple of slides here to show some of the tremendous growth we have in our research program here at the VA, especially in the last 10 years. You can see the number of our VA funded awards from precipitously, especially since we moved here and are co-located again with our active. So we now have upwards of 120 active VA funded research awards. So of course, when you get more VA research grants, you also get more money, which is a good thing. And so the last data on that graph is for this year, 2022, and we just surpassed $20 million in VA funding, and something we're very, very proud of. We're now one of the top 15 VA research programs in the nation based on VA funded appropriations. However, as we know, a strong VA research program does not exist in a vacuum. It takes collaboration with many, many partners. It requires engagement and support, not only of internal, but also external partners. These include the CU School of Medicine, of course, our academic affiliate, Covert, our primary IOB of record, and also our VA nonprofit Research and Education Corporation, the Denver Research Institute, who I also thank for helping sponsor this event and the Finlay Ahnen Lectureship. I would also like to thank Mr. Kilmer, our director, who I guess isn't here right now. But I'd like to thank Mr. Kilmer for his strong support of our research mission and helping us foster a culture of research here within the Rocky Mountain VA Medical Center, as well as the Eastern Colorado healthcare system as a whole, as we try to expand our research to reach underrepresented populations such as rural and frontier veterans. Finally, and I think most importantly, I really want to thank our research administration staff who have worked very devoted, tireless hours to help us keep up with this tremendous growth, even though the number of staff in our office have not kept up with our funding, but we hope to add additional positions this year. I hope people have had the opportunity to see the posters displayed in the concourse. They will be displayed all week, although today was really your chance to speak to the researchers themselves. The rest of today's activities. Next, we'll have some opening remarks from Dr. Vineet Chopra, the chair of the Department of Medicine at the CU School of Medicine. And then Mr. Kilmer, our medical center director. Following these opening remarks, we're very proud to have Dr. Finlay Macrae give our Ahnen lectureship this year. And then after Dr. Macrae's lecture, we will have several talks by our own local researchers. So you can hear in a little more detail about some of the important work that we're doing here for research for our veterans. So thank you all for attending. Please help yourself to some snacks and some modest refreshments. I would now like to introduce Dr. Chopra.
Good afternoon, everyone. It is such a privilege for me to be here today to be part of this important research day to represent the Department of Medicine. The day is especially meaningful to me for a number of reasons, but the most important one I will say is that I have been very influenced, and in fact, I would say molded by the strong VA culture and the culture of research in the VA. As some of you may know, I went to residency at the Mount Sinai Hospital in New York City, and my very first rotation in medicine as an intern was at the Bronx VA Medical Center. I can tell you a lot about that day, but I will tell you three things that still I remember to this day. Number one, the fact that everyone there focused on one mission, and that was the care of the veteran in front of you. Number two, the principle of using data to inform practice in every sense of the word data possible. And number three, the compassion and the empathy and the energy that staff and faculty brought, no matter what it was, if it was clinical care or research or even education. In fact, I was so taken by this culture that following my residency, I elected to stay on an additional year and do a chief year at the Bronx VA Medical Center, where I continued to experience that joy in caring for patients as well. Over the past 15 years in my prior posts, I've remained very close to the VA. I've been a VA-funded investigator, done clinical rotations at the VA in Ann Arbor, and I've remained a 5H-funded HSR&D investigator. In fact, this is the first time that I've actually been without a VA connection in my career. But I'm looking to change that, as Dr. Rice and I were talking about as well. I will tell you also, I have been mentored by VA investigators throughout my career, so I'm very grateful to all that the VA has done to me and has done for many like me in terms of our careers. What is unique and powerful about VA research is that it is inherently driven not just by scientific curiosity or the pursuit of data or the next paper or the next grant, but rather this burning desire to improve the care of the veterans we serve. Walking briefly to the poster hall this afternoon with Dr. Rice, that was full on in display in terms of that core value. Whether it was thinking about novel therapies for cancers that are prevalent in veterans, telehealth-related platforms to improve access to care in rural areas, interventions aimed at improving mental health and veteran safety, or even novel technologies to assess infectious and non-infectious diseases. I was delighted to see the unique blend of research that is so typical of the VA live strong in these roles as well. The theme of your research day, Science in Service of Veterans, is not only fitting then, but it was on full display today as well. As chair of the Department of Medicine, I am committed to building upon our very strong relationship with the VA. We share over 130 faculty here, many of whom are completely committed to improving the lives of veterans. I should also say that the research on display today isn't just about focusing on the VA, but the lessons learned will also carry forward to the university and to the department and beyond as well. I'm grateful to Dr. Rice for your leadership for today's activities. Also to Drs. Rebecca Scalzo and Amy Keller, faculty in our Department of Medicine. I have you to thank for being here today. Thank you for reaching out and inviting me. I know you spent countless hours organizing this, and your labor of love is visible and palpable. I look forward to continuing to support the VA in all of its missions, clinical research and education of tomorrow's doctors. And I assure you of my strongest consideration in the many years ahead. Thank you all very much.
So much. I have to fill in a little bit, some jokes. Here. Again, just so other folks know, maybe the size of our research program. We have about 180 principal investigators right now, most of whom are dual appointed faculty with the school of medicine or the Anschutz campus. But we also have other ties with organizations such as National Food Health, C. Boulder, CSU up in Fort Collins. And I think when we look at some promising future opportunities such as the PATH Act, I think we need to leverage our relationship with not only our closest academic affiliate, but these other institutions. So again, we can leverage our mutual scientific expertise, break down barriers where we're each working in our own lab or on our own study, and create more of a collaborative environment where we are truly focused on the end point, which is improving care for our veterans. And I was speaking to others at the posters this morning, and it's great to get research grants. It's great to get money and publish lots of papers, and you get a cell publication. But what does it mean if we're not changing how we care for our veterans and implementing this research into improved care and a better quality of life for our veterans? So as deputy ACOS, that's one of my big goals in the next several years, is to help break down these barriers. And I especially think for our career development awardees, I guess I have a big part there because I was one. So I'm a little selfish there. But I think at that stage in career development, starting those collaborations early. So say if we have basic scientists, having them work with health services researchers, in the beginning to make the research more clinically relevant, more likely to lead to actual changes in our clinical care. Other major improvements that we need to make here, while building up our research administration staff. I think I mentioned that earlier. Two main areas we really need to attack. One is protocol approval time. We're letting our investigators down by not getting protocols approved through our R&D committee quickly enough. It's not a matter of our hardworking, dedicated staff. It's a matter of those graphs that I showed you. We just completely outgrown our resources. We're sort of a victim of our own success. So the good news is the addition of the deputy ACOS position. We have already an additional position in our R&D committee group. We plan to add at least 3, if not 4 additional positions, both in R&D committee as well as financial support. That's the third area we're really, or the second area we're really failing our investigators. We just don't have enough purchasing support to keep up with the amount of money that we administer every year. Does anyone have any questions while I try to? I would like to say that the talks that were chosen after Dr. Macrae's, I think, really illustrate the bench to bedside concept on the bedside of things, and I think that's why these talks were specifically chosen, so you can see how this important VA research culminates in changes in clinical care. Since I don't tell a lot of jokes, I'm just gonna stop now. Please help yourself with some snacks. And I think Mr. Hillman will be here in a few minutes. No, yeah, I know.
Yeah.
This talk will be good. And you can kind of see I was.
Oh, yeah. I appreciate it.
I did not have that same. I was like, just take a picture with us.
Seagull.
So I'm standing there in front of the audience.
Oh, yeah.
And he's like, "What?" I'm like, "Dude, I texted him."
He's got fresh air, dry brain.
That's right.
Yeah.
Taylor would have been on top.
Yeah.
She'll get the next hour.
Great. Yeah, really,
Okay,
nothing else.
and then you know what word I can never say correctly.
Yeah, it does sound, it actually, like you need to
I wish I could just say that cancer, but
It's
it's
not.
colorectal. I always stumble on that.
Right.
Always.
Yeah, I will call in case.
Yeah.
Like, do we know about? It's gonna come in the video. It's like, not-
I don't really shouldn't be that long.
Okay.
No, I think about 15 minutes.
Yeah.
That's-
Yeah, we're gonna post this also.
Yeah.
All right, everybody, sorry for that, story, but I'm here to introduce now our fearless medical center director, Mr. Michael Hilton.
This one or that
Both.
one? Okay. I don't know anyone that apologizes for extra break and networking time, so hopefully you made the best of that. But I just wanted first, thank you for coming together. And just a huge round of applause for all the poster board session participants. That amazing work out there. So thank you very much. So today we are recognizing some of the incredible work that VA researchers are doing to advance healthcare. And I could not be more pleased. Wow, that's the wrong page. Sorry. Okay, hopefully these are in order now. Let's see. So as you know, VA has 4 core missions: 1, clinical care; 2, education; 3, research; and 4, emergency management. Research is a crucial element. And for me, this is something that touched me early on in my career. I'm very grateful for the time that I had as an intern at VA Puget Sound, and then as a new VA employee at VA San Diego. And there was a researcher there that I just frequently will mention, Dr. Julene Baker, who's a psychiatrist, because she really instilled in me as a new clinician, as a baby social worker back in the day, the importance of effective clinical screening to feed into effective research. So research supports clinical, clinical supports research, and that has always been something that has stuck with me. So I'll forever be grateful for my time that I spent in California and Washington State and learning from those great institutions out there. I'm so sorry, these things got out of order. Okay, I think it's all right. Today, we are recognizing some of the incredible work that VA researchers are doing to advance healthcare, and I could not be more pleased to be standing in front of all of you to kick off the next series. For Research Days, we have made it a priority to feature our Dennis J. Ahnen visiting lectureship to honor his mentorship and his overall achievements in disease prevention. Dr. Ahnen was an international expert in the field of colorectal cancer. He also achieved the highest honors in his field. He had also served 32 years in the VA and exemplifying VA's core values and instilling in future researchers the value and impact of research has across medicine. Dr. Ahnen may no longer be with us, but his memory continues to live on through the lectureship with our VA Eastern Colorado healthcare system. I'm thankful for many of our researchers here at VA Eastern Colorado We continue our goals of being one of the top 10 VA research sites. I'd like to share a few accomplishments that show how we are achieving these goals. In the last 2 years, we have advanced from 22nd to the 15th largest VA research facility. We were recently selected as a VA node site to serve as the VA sponsor site for typically smaller and rural sites who aren't historically involved in research, but now can be because of our support. We're also the new host site for our National Vet Home Program that focuses on environmental hazards. And I'm hopeful that that will lead to more research funding coming in. We have increased our administrative and operational support to include increased business controls and adding Dr. Pam Rice as our very first ever deputy ACOS of research. So applause for Pam. Thank you for joining us. But none of this would have been possible without all of your dedication. And to highlight some of that, I'd like to highlight a few of our team members. These efforts make VA Eastern Colorado stand out across VA research. The research being conducted here is having incredible impact, and I would like to highlight a few of the studies. First, Dr. Jacob Siegel, is he with us today? Oh, okay. Dr. Jacob Siegel's work providing prosthetics limb sensory feedback for upper limb amputees. Dr. Andres Vasquez Torres and his research on antibiotic-resistant gram-negative bacterial infection, which are associated with high rates of morbidity and mortality around the globe. Is Dr. No? Okay. Dr. Silpa Kraft's work on identifying clinical markers and monitoring veterans of post-9/11 deployment lung diseases associated with exposures to burn pits and desert dust, which have been linked to post-9/11 deployment-related asthma, small airway lung disease, and bronchitis and other chronic illnesses. And finally, Dr. Lindsay Monteith and her work on female veterans within VA family and working together to prevent firearm-inflicted suicides among female veterans who are twice as likely to die compared to their non-female veteran counterparts by lethal means. You can see how this research directly ties to and with social and health challenges, specifically to our patient population, and ultimately we'll be able to see the impact that this research has across the world. Thank you to all of you for being here with us for this work that you do across your profession. We are honored to have Dr. Finlay Macrae as our next speaker. Dr. Macrae is a professor and head of colorectal medicine and genetic program at the Royal Melbourne Hospital in Victoria, Australia. His clinical work and research in cancer genetics and prevention covers early detection and prevention through designing and implementing randomized control clinical trials. Dr. Ahnen was a colleague and collaborator with Dr. Macrae, co-authoring several seminal publications in familial cancer risks and prevention. Today, Dr. Macrae will be presenting a culmination of his work from other leading scientists, including Dr. Ahnen, titled Curating the Hereditary GI Cancer Genome, a Herculean Task. Please welcome Dr. Macrae.
Thank you, Mr. Kilmer, for that generous introduction. I very much value this opportunity to address you today. Very much feel honoured and privileged to be here to be presenting you the Dennis J. Ahnen lecture for 2022. So my task is to alert you to the curation of the hereditary GI cancer genome and the Herculean task this represents. And I think you'll understand as I address you through this, this is really a global exercise that needs the world's resources, intellectual and otherwise, to address the problem for the benefit of human health. Oh. So Dennis is indeed a great friend of mine. We used to meet regularly around posters of Digestive Diseases Week all around America each year, and we would tend to congregate around posters of common interest. This led to a very deep friendship that we felt between the two of us, and we've shared that over the last couple of decades. Indeed, over the last 4 years, we've had weekly conversations with the Ahnens and to exchange news and views of our families and our terms of Dennis's, our mutual resource, until, of course, Dennis sadly passed away just over 2 years ago. I like to draw your attention to two things on this slide. One is the DFOF at the top there. Dennis actually introduced this nomenclature to me, this postnomial. And when I saw it first at some DDW presentation, I wondered what it was, but he explained it shortly afterwards. It represents dear friend of Finn's. So indeed, when you saw my titles, you would have seen there DFOD. So that represents the strong bond that I shared with Dennis over many, many years. Indeed, this is a paper that I did with Dennis. It's one of the only papers that had both of us as the only authors. So that's why I put it up specially. But indeed, we were co-authors on perhaps a dozen papers over the last couple of decades. This one I think is special because Dennis was a great person for words and for concepts. So we drew this together, and it was about describing the possibility of an accelerated carcinogenesis on the background of a base excision repair defect inherited, now known as a MUT-YH-associated polyposis condition. But you can see we played on the word myth and MUT-YH to come up with this title of the hare, the tortoise, or is it a myth, with respect to the carcinogenic process on this DNA background. We did share great friendships, and this was just a few years ago at Dennis's 50th wedding anniversary. And you can see that Dennis and Carol reaffirmed their vows with me there as the cleric in the background, offering my authority over their reaffirmation of their wedding vows. So I want to move on to the topic. And all of you, of course, are aware that we're in the middle of a genetic revolution that's being applied to medicine. And that's no more obvious in cancer, no more obvious in any field than it is in cancer genetics. This is a little out of date now, but it's meant to represent the fact that, of course, now we do panel testing, testing for multiple genes in different clinical scenarios that is secured at a cost that is substantially less than what we were able to do just 5 or 6 years ago, when our testing techniques were based on Sanger sequencing on single genes. So the cost has rocketed down in terms of our capacity to do germline testing, and all of this talk is going to be about germline testing, not somatic testing. And it does, however, bring into the challenge that we often get off-target genes that are being tested for, off-target for the clinical condition that we're faced with. And that leaves us with a big problem in terms of DNA interpretation, because we indeed get a lot more variants of uncertain significance that trouble us in terms of clinical application and genetic counselling. Now, I'll take an aside here that my interest in this was initiated about 12 years ago when a scientist with whom I worked at the University of Melbourne called Professor Dick Cotton recognised that the problem with the application of genomics and genetics to human health was not going to be the capacity to sequence, but the capacity to interpret the sequence. And that has proved absolutely true. We've now got enormous capacities for sequencing, but we're left with the major challenge of interpreting the sequence often. So he formulated the notion of the Human Variome Project, which was simply the idea of centralising all sequencing information from all humans, from all countries, with all diseases on one central database. That was the notion of the mission of the Human Variome Project. And indeed, a central database was established on a server in Holland, which is known still as the LOVED DNA database, which stands for Leiden Open Variant Database. And that's been assembling information about DNA variation for the last 15 years. And the idea that Dick Cotton had was that where there was a challenge in terms of interpretation, what you needed to do was annotate the variant with various bits of information, which I'll describe, to enable us to achieve interpretation of a particular sequence. And most challenging, a SNP, a missense mutation, which can be very challenging to interpret. So the Human Variome Project initiated that concept. I must say that it took about 8 years for our colleagues here in the United States to realise there was a problem with DNA interpretation. And it was a big challenge. And although Dick's mission was global and included United States investigators, it was mostly European that picked up the challenge in those earlier years. And we looked at it from Australasia with interest as the science moved and developed between the United States and Europe. But the Europeans were leading the way in terms of understanding this challenge until about 8 years ago, when the National Institutes of Health really stepped up and recognised we have a problem. And to the great credit of the United States and to the benefit of the world, they put money into the task of DNA interpretation. So that was the birth of ClinVar and subsequently ClinGen. So ClinVar is a similar concept of centralising DNA variation on a central database in Washington. And ClinGen is the overall structure of the committee structure that oversees the variant information that lands on the ClinVar databases. So we have a ClinVar database that sucks in DNA sequence information from all around the world, but particularly the United States and particularly private laboratories in the United States to centralise the DNA variation with some associated information. And we have well-organised committees, they're called variant curation expert panels, that are ClinGen organised to oversee the information that's landing on the ClinVar database. The ClinVar database accepts not only the sequence information, but the interpretation of that sequence information as derived from the submitting laboratories. So you will see a variety of interpretations for the same variant landing on the ClinVar database from labs around the world, including diagnostic labs in the United States. So I'll mention, and you can foresee that there's a challenge there and there's a problem, which I will come to. So we do get a lot of information. And one of the differences between the LOVED database and the ClinVar database is that the ClinVar database is more modern. It attracts information from panel sequencing, and it therefore has a lot more variants of uncertain significance from off-target genes that land on the database. The LOVED database was largely driven from clinical sources that were sequencing clinical syndromes that had definite phenotypes and were much more likely to have pathogenic variants because of the data source that was being driven from. So we now know, of course, that uniform germline sequencing of cancer patients will return a substantial number of pathogenic DNA variants in just about every cancer that or series of cancers. And they can be both on-target genes and off-target genes. So here's a paper now that's about 8 years ago from Matt Yulgelchen, who was analysing the outcomes of uniform DNA sequencing in patients presenting with colorectal cancer. These were patients that were suspected to have a germline mutation, so you'd expect that the frequency of mutations would be higher than a more representative sample of colorectal cancer patients. And in the top diagram there, you can see that of the mutations that were identified, and not shown here, but that was about 12% of the patients that were sequenced, 12 or 15% actually had germline mutations. And about just over half of them were the known Lynch syndrome mutations. But there are also a lot of non-Lynch syndrome mutations identified, represented in the lower pie charts there, and the non-Lynch syndrome mutations were not only ones like APC and MIT-YH, but they were quite off-target genes like BRCA. So you wouldn't expect to be finding BRCA gene mutations in a cohort of Lynch syndrome patients, but you do find them, and they contribute to that overall 12 or 15% of mutations that you find even in an unselected series. And then there are moderate penetrance genes that represent another proportion. So uniform germline sequencing of cancer patients is really just about on us now, when all patients presenting with cancers, and colorectal cancer in particular, will have germline sequencing, because there is a substantial pickup of autosomal dominant or recessive or moderate risk genes. So what is the importance of curation? Why do we have to have curation of all this information that's being centralised on databases such as ClinVar, LoVet? Why do we have to have variant curation expert panel? Why do we have ClinGen? Why do we have to have ClinGen? Why do we have to have had what preceded ClinGen, and that was, in our case, the International Society of Gastrointestinal Hereditary Tumour Variant Interpretation Committees? It was because of the risk of misclassification. Clearly, if a DNA sequence is interpreted and it's wrong, that can have major consequences for clinical management and genetic counselling. Probably the most outstanding example of that might be in the BRCA field, where if a BRCA mutation is identified and it's wrong in terms of its classification, for example, it's classified as pathogenic when really it's not pathogenic, and that individual goes ahead and has a bilateral mastectomy, and their family through cascade testing are identified and may have bilateral mastectomies. If the classification's wrong, that's a major issue in terms of mismanagement clinically. So there is an incidence of misclassification for a variety of reasons. That is relatively easily identified where different laboratories submit different interpretations of the same variant onto a database such as ClinVar. So if you look at the ClinVar database, you can fairly easily identify on the basis of the different labs that are submitting their interpretation of the variants they find, you can identify discordances. And that represents the first line of investigation for committees such as the ClinGen variant curation expert panels. It leads to inconsistency in genetic counselling. You could imagine that if a variant is found in one branch of the family and it's interpreted as pathogenic, and elsewhere, perhaps in the same family, sequencing is done and it's interpreted as benign, the families get totally confused by the differential advice that's quite different. So this, I think, is a really important feature of clinical care that I particularly have been involved in ever since the days before ClinGen and ClinVar. Here's a representation, a recent representation of the challenge that we face in terms of seeing consistency of interpretation of germline DNA variation. Here's the genes that we're familiar with in hereditary GI cancer. And for example, if you look at the top line there, when this when this table was first put together about 6 years ago, there were 1,860 discrete APC variants on the LOVED database, and no, other way around. About 6 years ago, there were 1,775 discrete variants on the LOVED database, and in terms of ClinVar, there were nearly 7,000 variants, but now the number has increased to almost double that amount, particularly in the ClinVar database, which is sucking in vast amounts of information from particularly private labs in the United States. And you can see, if you move along the column there, in terms of the APC mutation, APC gene, there are just over half of them are either variants of uncertain significance, or there are conflicts in interpretation between different laboratories submitting their assessment of the pathogenicity about the same variants. And you can see, however, that there are quite a, if you move to the next column there, you can see that the balance of the variants, that's the difference between 100% and 57.6%, there's 42.4%, do have classifications that are either pathogenic, likely pathogenic, likely benign or benign, which is more secure, if you like. We're a bit more comfortable with those, but still there could be errors there. And more than half of the submissions are coming from a single submitter. So there you've got very little capacity to draw out the possibility of discordance or wrong interpretations. And you can see there for the rest of the genes, similar figures. So how do we address this? Well, we did indeed establish what we call the Insight Variant Interpretation Committee. Before ClinGen and ClinVar were constituted, and we addressed this particularly around the variants landing on LOVED, but with the entry of ClinVar and ClinGen into the mix of international expertise, we put ourselves forward, really, and were accepted by ClinGen as being the expert variant curation expert panel that that operated to oversee this issue of discordant variants and misclassification as landed particularly on the ClinVar database. We are quite a big organisation. This is just one subcommittee of Insight ClinGen variant curation expert panels that address in particular the mismatch repair genes associated with Lynch syndrome. And if you look down the list there, it truly is a global international effort. There's representations from countries and continents all around the world who really do this voluntarily. They put up their time and their effort to participate in this task for ClinGen of interpreting the variants as arriving on the ClinVar database. There's about nearly 50 members there that we coordinate. Actually, from Melbourne, we coordinate this effort to make sure the variants in the mismatch repair genes and actually in the other genes are as accurate as they can be. So how do we do it? Well, it's fairly easy where the variant is obviously truncating or frame shift, or there's structural variations such as large deletions or interference with initiation codons. But it's not so easy where it's a missense variation where you haven't got that biological understanding that the change is damaging or pathogenic. So what we do is we look for annotated information that relates to each and every variant in different domains of information. And much of this talk now is going to be about that process of analysing through different domains of information the data that is available on each and every particular variant. So first of all, there is the sequence base, and that's just what I've described, but largely the missense then lead us into this task of applying other domains of information that is collected by physicians and researchers, either sitting in their filing cabinets and not available to the world, or published, or just being submitted to the database without any necessarily published information in the general literature. So there's segregation information, and that can be very powerful. I often describe the power of that and the utility of a global database and a global approach to this by pointing out that if a particular variant in a family, a missense variant in a mismatched repair gene is found in a Chinese population in Beijing, they have in the main one-child families, and they have very little capacity to do segregation analysis. However, if the same variant is found in Rome, in a culture of a Catholic background, they have large families, and they have the capacity to do segregation analysis and come up with very powerful data, probability data, but still very powerful, that a particular variant is pathogenic and associates with the disease in the family. So unless you centralise that information on a global database, the Chinese aren't in the race because they can't get access to the information. So it's very important to share the data and to do it optimally, and as we're doing, through a globally accessible database. And to its great credit, the NIH, the NCBI, and the NHGRI do have a global perspective about this, and they're not at all territorial about the information from where it's derived, or indeed restrictive about where the funding is applied, because this truly is a global humanitarian problem that needs to be addressed globally. So there is segregation information that can be put together in a probability perspective, because that's what LOD scores do. They give you a probability, an association. In the case of Lynch syndrome, we can derive extra information from the observation of microsatellite tumours occurring in families. So even if you can't do segregation within a family, you can certainly look from a series of individuals or families, the proportion of tumours that are microsatellite unstable versus stable, and again come up with a probability that a variant associated with the people with those tumours is pathogenic, causing that microsatellite instability or not. Even just a missense variant, you can make that association, and it gives you information. More recently, we've used allelic frequency in the populations that we're addressing, and simply this means if you've got a rare disease and you've got a missense variant that's appearing, and you're trying to work out whether it's benign or pathogenic, if that variant occurs more frequently than the incidence of the disease in the population, then it's probably not pathogenic, because you'd be seeing more of that disease in the population that was pathogenic. So you can work out statistics around this, and you can draw in thresholds so that if the variant you're finding is much less frequent than the variant in the ethnic population you're looking at, in the you can put extra information in to help in the interpretation, and you can quickly dismiss a whole lot of variants that might be found in families with a particular tumour phenotype, but if it occurs frequently in the general population, it's not going to be pathogenic. So that's a powerful way to push variants from uncertain to benign, just on the basis of allelic frequency. You can also look at a co-occurrence in trans. So occasionally you find a variant that represented there with the question marks that you're uncertain about. But if in trans there's a definitely pathogenic variant causing the same phenotype, then you can make the conclusion that the question mark one, the one that you're wondering about, is probably not pathogenic, because you can explain it on the basis of a definitely pathogenic in trans in that individual, and that adds information. Then there's the domain of experimental data, and there's now about 50 different functional assays for the mismatch repair that can be done in vitro, sometimes in live systems such as yeast, where typically a variant of interest is inserted into some biological system, and then some measure of mismatch repair capacity is the readout from the assay. And you can use that information to help interpret whether a particular missense change is causing mismatch repair, is correcting the mismatch repair or not. And then there's a whole lot of in silico processes that can be used by clever computational biologists who assess whether a particular amino acid change that might be generated from a DNA change causes a three-dimensional change in the structure of the protein, which disrupts its function. So that's other information that can be added. And finally, there's evolution and conservation, where the sequences of all sorts of forms of life from very primitive to humans are now well known. And in the case of a particular gene, here we're talking about mismatch repair genes, if a particular portion of the gene is conserved from the most primitive forms of life to the most advanced forms of life, if a DNA change is seen in a well-conserved proportion of the gene, it's much more likely to be pathogenic than if the DNA change is seen in a variable portion of the gene from primitive life to humans, because the function of the gene still works just as well whether it's primitive or advanced. So clearly the bits that are conserved right through evolution are important for the function of the gene. And that's another piece of information that can be used to help interpret these difficult DNA variants. So all of that information is assembled in interpretation, often in a Bayesian likelihood analysis approach. So mathematicians tell us through Bayes that you can multiply the probabilities together on each of these domains of information, even though they're quite different in terms of their nature, and you can come out with a much stronger probability about the relationship of the variant to the disease. So that's a core function of variant curation and understanding, which is applied variably by different laboratories and different settings to the task of interpreting a variant that might be before a particular lab. So one of the advantages of FlinGen is that they've introduced a standard way to approach this. This is the American College of Medical Genetics criteria for interpretation of DNA variation, and that has become the default now in the world of criteria to apply to interpreting DNA variation. So the ClinGen process, which is what this is representing, now has variant curation committees in all sorts of diseases, and you can see hereditary cancer there in the middle is one of them, and our particular patch of hereditary GI cancer is a subset of those, and a subset of those is the mismatch repair genes that fall into that process. They come up, the criteria there, you can't read them and you're not meant to, but they're up there in the top left-hand corner. They're structured criteria. They're based largely on what I've just described in the last sentence, but they apply not only the domains of information, but they give you a guide as to how much weight you can put on different parameters and include that in the mathematical calculation about interpretation of the DNA sequence. And I'll describe that a little bit more. They do allow, and this was only recognised some time after the ACMG criteria were defined, that you can get more bang for your buck, more mileage, if you approach the task from a disease and gene-specific basis. So you can get better effectiveness in classification if you take on board gene and disease-specific considerations. For example, in the last slide I just showed you, we took on board the information about microsatellite instability in the cancers. Now, clearly you can't use that information for cardiac gene analysis. It's specific to the gene and the syndrome involved, but it's also highly relevant to the process of DNA interpretation in those genes. So there is a capacity that ClinGen allows to modify the criteria in a gene and disease-specific fashion to enhance the capacity to interpret the DNA sequence. So I've explained this largely already. This is about the history of how all this occurred. The time before ClinGen and ClinVar were on the scene from 2012 to 2019, the rules that the Insight Committee came up with, which were similar but not the same as the ACMG criteria. And currently the agreement for our Insight Committee to become the Insight ClinGen colorectal cancer polyposis variant curation expert panels, which will recognise even now as more dominated by European expertise and Australasian expertise than US expertise, but there has been a lot of US expertise that's come in since the ClinGen process developed. And the utility of the ACMG framework has been broadly recognised because of its flexibility in terms of combining different evidence combinations with different levels of strength. So it really has been important in terms of standardising the field and allowing consistent interpretation of DNA variation. So we've introduced new criteria influenced by the ClinGen recommendations. And the process involves, in order to qualify as a bona fide VCP, the appointment of duly qualified people on those committees. So all those people were mentioned had qualifications that enable them to participate. And they, in terms of the core function of biocuration, which is a big task, there's a process of training that needs to be undertaken by the core biocurators using the ClinGen interface tools and software tools to do it. This really just summarises it, the application of this whole process to the mismatch repair genes. There are large numbers of various strengths of information relating to pathogenic or benign criteria. You can see the P, it represents pathogenic criteria, the B, benign criteria, and different levels and strengths of evidence. You can see in the top line, strong, supporting, moderate, strong, very strong, can all be introduced into the equation to lead to a point system that gives you the pathogenicity. So the ACMG criteria, rather than being based on statistical probabilities, uses a point system which is a little easier to implement and use as in the biocuration process. But it uses the same, you can see, domains of information, allelic frequencies, tumour characteristics, co-segregation, etc. Not all the criteria that are in the original ACMG set need to be used for different genes, and that's a task that the variant curation expert panels address. Do we need to use all the criteria or only some of them? So 9 of them were not used at all, 16 remained unchanged, and quite a number of them were modified for gene-specific reasons to allow better classification for this particular gene. That represents the modifications. You can see those arrows change from the original ACMG criteria. That represents the modifications that have been introduced for the mismatch repair genes in particular. Some of it's quite complex to apply. This is in the case of loss of function variants from a genomic level itself, and whether the change is a nonsense or frame shift, which is a very high probability of being pathogenic, through to splice site variants, which may or may not have such strong relationships to the outcome. Deletions and duplications often are very strong, and interferences with initiation codons or codons at the 3' end of the gene. There's a limit in each of the genes beyond which a DNA change will not be pathogenic because it only clips off the end of the gene and doesn't involve interactive sites with other genes along the pathway, so they can be excluded from pathogenicity. That's what that box towards the top right represents, and it's different for every gene. So this again is a gene-specific modification that's applied in the case of mismatch repair genes. What we do as we go through the qualification process with ClinGen is take a pilot set of variants, usually variants that are selected to be difficult to interpret, and apply our modified rules to those variants. In this case, 45 variants were selected for this task and classify them according to the modifications we make to the ACMG criteria. In this case, of the selected difficult ones, 17 were returned benign, 15 were still uncertain, and 13 were pathogenic. This information is fed back to the overall sequence variant interpretation committee, an overriding committee of ClinGen, which oversees the work of each variant curation expert panel and decides whether their approach is bona fide and can be accepted as the authoritative process for DNA interpretation for that particular gene. Here the pilot variants have been fed into the ClinGen curation interface, and this is the way it gets submitted through ClinGen to ClinVar. And you can see that the various levels of evidence, be it moderate supporting towards pathogenic or benign, are assembled for each variant, and a provisional classification there in the centre is come up with on the basis of that combination of supporting evidence as to whether the change is pathogenic, still remains uncertain, likely pathogenic, or still remaining of uncertain significance, with not enough evidence to push it one way or another. And each individual biocurator has their own interface that expands on this by providing a little bit more detail on how an individual biocurator comes up with a pathogenic classification. And the process then, if there's inconsistency between independent biocurators using the modified criteria approved by the ACMG, if there's still inconsistency, that's brought forward to a teleconference for the big committee, which then come out with a consensus view about particularly difficult variants in terms of their interpretation. Those conferences occur about once every month or two. In Australia at 6 a.m. in the morning. In America, it's about 2 o'clock in the afternoon, and in Europe, it's about 10 p.m. at night. So it's a challenging exercise for all those people involved. This is an example of the functional assays. Here, a variant is selected. It's recreated by PCR. It's inserted into in this case, the variant was in MSH6. The variant MSH6 protein is produced in vitro. It's inserted into a MSH6 deficient cell line. A substrate is added, and then an assay of mismatch repair activity is undertaken, and that assay gives you a readout as to whether that variant is indeed pathogenic or not. This is one of about 40 different assays. They all test slightly different functions of the mismatch repair genes, but this one has probably the best runs on the board in terms of being useful and adding that domain of information, functional assay information, to the task of interpretation. But it's not the be-all and end-all itself. It has to be added to the other domains of information in that Bayesian likelihood analysis to come up with a conclusion. So this is work in evolution. In terms of the mismatch repair genes, where are we at after 2 years? Well, the committee's been selected, the biocurators have got to work, the pilots have been classified as we've seen. We're now actually at the point where all this information has been submitted to the sequence variant interpretation committee, which is, of course, a ClinGen US body who provide their approval or disapproval of the process. That's then published. It will be published and then deposited on the ClinGen and ClinVar database as the definitive process for and criteria for sequencing the variants. Before we had this point-based system, indeed we had a more mathematical Bayesian analysis approach, which instead of points came up with a percentage probability in a classical Bayesian approach, multiplying the different probabilities from the different domains of information. And if the probability comes up as greater than 99.5, that is described as definitely pathogenic. If it's 95% or more, it's likely pathogenic. At the other end of the scale, if it's 0.5% likely to be pathogenic, then it's called benign. And if it's 5%, it's likely benign. And everything in between is still very of uncertain significance. So what we did was apply the Bayesian approach, and Sean Taptichian from Utah has been the champion of this sort of approach in interpreting DNA variation right across the genome. But he has focused on cancer-related genes. So we compared the Bayesian statistical approach with the ACMG points-based approach to see how much of a difference there was, even with these high-level approaches to DNA interpretation. And here is the same set of 45 variants in the mismatch repair genes, where most of them align, whether it's, you can see the ACMG approach or Bayesian approach, almost all of them come out with the same information. There are a few that are discordant between those two approaches, where you can see the top line there, only 3 actually. The Bayesian approach reached pathogenic, while the ACMG criteria did not have the same evidence used. That suggests that perhaps we should be updating the criteria, the ACMG criteria, and look at why that occurred. The next one was an ACMG used one particular level of strength of evidence, which was a type of evidence not used in the Bayesian analysis. And therefore, there's room for consideration of how we approach that. And the last one was the variant curation expert panel determined that Bayesian benign was not appropriate due to lack of clinical evidence, and changed the ACMG criteria. And we changed it to be stronger with the ACMG criteria. So these are all helpful analyses as we go forward to make sure we're fine-tuning and got the classifications right. So the ACMG criteria have been useful in the field. They clearly defined the evidence. They established thresholds for example, allelic frequency, probabilities in terms of in silico analyses. They give thresholds for functional assays, and they give better guidance on the strength of information that segregation data provides. It is the de facto classification standard adopted globally and acceptable for clinical genetics. But nevertheless, the older Bayesian approach and the ACMG approach are broadly compatible, and both approaches inform the other. Now, I just want to move on, and this is the committee and acknowledgements that I would provide for the core group of people that are represented in that mismatch repair curation process. I want to touch on perhaps more quickly the APC gene. So the NIH grant was the terms of reference for applying for the NIH grant to support the curation was that the genes that would be funded would be those that were less mature in terms of their interpretation processes. Now, the genes that were most mature in the cancer space were the BRCA genes and the mismatch repair genes. So they were judged by the NIH as not requiring extra funding to carry through this process of curation. But there are a whole bunch of genes that were really quite immature in terms of the curation exercise. So actually the NIH grant only funded the APC gene, MUT-YH, the hematomasis genes, POLE and POLD1 to carry through this curation process, including the establishment of variant curation expert panels. So the APC gene was the first gene funded under the NIH gene to get legs and run in terms of putting this on the map. The funding has been available for about 18 months, and this has run very quickly and well through the international collaborations involved. Again, this is the Insight ClinGen APC variant curation gene process, and it's addressing the APC gene that you can see represented there. We know a lot about the genotype-phenotype correlations with the APC gene. And of course, its association with the phenotype of familial adenomatous polyposis as being the most frequent adenomatous polyposis condition with a high risk of colorectal cancer. And we know that pathogenic variants are found in about 80% of FAP families and 20-30% of attenuated FAP families. The same process, you've seen this diagram before, is applied to the APC gene, but here now we have the NIH ClinGen, NIH-funded variant curation expert panels formulated across all those other genes, with co-PIs being Mark Greenblatt from Vermont and Matt Ferber from the Mayo Clinic, who oversee the whole process of all these genes, but each of the genes have members that represent different domains of expertise, from clinico-pathological expertise, experimental in vitro expertise, molecular diagnostic expertise, statistical expertise, and computational in silico expertise. There you can see some of the committee members for the APC gene that are working hard. Some key North American members of that committee, as Sharon Plon from Baylor in Texas. She's really a key member of this whole process of DNA interpretation, particularly in the cancer genes. Deb Ritter, also from the Mayo, from Baylor, is a very active member. Mark Greenblatt from Vermont is a wonderful molecular clinical geneticist who has great reach, and of course is a co-PI of the whole exercise. And the other, and Sherry is a student of mine who's now really pretty much the main drive in the APC gene. She's just had 6 months in Germany working with the chair, Stefan Oretz from Bonn. And we go through the same process of working out modifications to the criteria that are applicable to the APC gene, developing biocurators and their expertise to address it, coming up with pilot variants which are submitted, which are judged against the modified criteria, feeding that back to the overall ClinGen sequence variation interpretation committee. And this committee has only last week had the final approval from the variant interpretation working panel of ClinGen to apply their modified criteria right across the whole 9,000 variants in the APC gene in a definitive fashion, no matter what the submitters say from the individual labs. Here we go. So we're moving forward, looking at the other genes, the hematomatous genes, the poly and poly-1. The mismatched rare ones in italics because it's not NIH funded. And the different domains of information are broken down, as we've mentioned. On the top there, you can see the PIs of the different domains of information from around the world. And there are the principal investigators, gene by gene, that head the variant curation expert panels relating to the different genes in our domain, and again underneath the application of the different expertises to all of those genes. I'll move on from that. So there you can see the different domains, again, the same depiction of the ACMG criteria that are APC gene modified in this case, and according to different strengths of information relating across the top and the different domains of information relating down to the right in a matrix that is applied, and those different levels of evidence that are represented by those codes are assembled, and according to their strength and the different domain, they can be ascribed in combination, and that combinations can be flexible to lead to definitely pathogenic, likely pathogenic, variant of uncertain significance, likely benign or benign. And this, of course, again in a gene-specific fashion, applying the modified criteria. This represents the APC gene modified criteria, bringing in APC gene-specific elements, and that's probably most notable in terms of applying the clinical phenotype to enable a classification, bringing in clinical information to support the pathogenicity of a variant that might be found in an individual. So that adds to information from other domains, which allows, again, the final classification with the combination of those levels of evidence represented by those different coloured boxes. It's quite complicated and it needs some experience to do that. So here we have a depiction of the variants that are on the LOVED database, derived principally from people that have got definite phenotypes, and the variants that have been identified by the type of variant, frameship down to structural variants, and then the assessment of pathogenicity of those variants on the LOVED database, from pathogenic to benign, with variants of uncertain significance in the middle. And the red here represents the definitely pathogenic variants. And you can see that 92% of the variants on the LOVED database derived from people with definite phenotypes are identified as pathogenic on LOVED. Whereas if you look at the same information on ClinVar, you can see the vast majority of variants of uncertain significance, with only a small proportion, 11%, being classified as pathogenic or likely pathogenic. And that is because of the derivation of the sources of information. The ClinVar database is attracting all this sequence information from panel sequences, from off-target assessments, i.e. they're testing genes that are off-target for the phenotype, and therefore much more likely to come up with variants of uncertain significance. So the problem in terms of variants emerging from this part of the world is much greater than other parts of the world, who generally, at least historically, were only submitting information from on-target phenotypes for classification. Sherry, in terms of APC, has developed an algorithmic process to enable this whole interpretation to occur more quickly. Here is a breakdown of the proportion of variants that can be classified according to different classes of information. You can see it's quite complex, but you can run through an algorithm and you can come to a conclusion somewhat more quickly by applying this algorithm, using as the basis the modified ACMG criteria to reach a conclusion about pathogenicity. The simplest one here, as I mentioned before, is if a variant is if a variant that's asserted to be pathogenic is found much more commonly in the normal population, it's not going to be pathogenic. So you can immediately dismiss that and put it into a benign category. So that's one of the first steps in the algorithm, to cut down something like 30% of the variants in the ClinVar database into a non-pathogenic category. It still leaves you with about 780 that require detailed consideration just in the APC gene. When you apply the APC criteria to the pathogenicity that is on the InSight database and compare them, you can actually come up with quite a movement of variants one way or the other by applying the ClinGen criteria, the modified ClinGen criteria. Some of them can be quite troubling in terms of their movement from definitely pathogenic to definitely benign. So those are circled there, and they require a lot of thought. How do we manage a family where a classification has moved from definitely pathogenic to definitely benign through a change in the criteria? That introduces enormously complex counselling issues to the families that have been counselled on previous applications of either outdated criteria or classified at times when not all the information is available, as is now, to enable a better classification. So I've nearly finished. This is probably representative of the work that Sherry's been doing recently to enable this process to occur and reach a conclusion more quickly. I just want to move on to the hematomatous genes. I've been privileged to have a whole range of students who've addressed their research periods to times that support the task of classification of the GI cancer predisposing genes. This is one of my students just recently completed her year, who's assembled the world's information about DNA variation in the BMPR1A gene, which is one of the genes that causes juvenile polyposis. And the importance of this work is that it associates definitely pathogenic variants in this gene with different phenotypes, and there are different phenotypes in juvenile polyposis. They're represented by the different colours here. But if you understand this variant to phenotype interaction, then you can use that information to classify variants of uncertain significance in the gene. So this is just one of the domains. This is one of the efforts to support the process of classification. There hasn't yet been a variant curation expert panel mature enough to consider this data, but this will feed into that committee once it starts working early next year under the NIH ClinGen process. So what's the future of this? All of this information will be published over the next 6 months that I've described as to the work and the outcomes of modifying the criteria after the approval of the ClinGen Sequence Variation Interpretation Committee. Then there will be work that address the much larger number of variants that sit on the database beyond the pilot variants, 9,000 in the case of the APC gene, using those algorithmic approaches. And there'll be prioritisation of variants that are of concern to the clinical community, those that still have discordant interpretations from different laboratories, those that are addressed to the variant curation expert panel because something's cropped up in a family that has major clinical consequences for accurate interpretation and there's not enough information available. The centralised committee is in a position to draw their resources from across the world to see if they can draw out information from hidden filing cabinets that relate to variants in those particular families to help the interpretation. And as a ClinGen variant curation expert panel, we have the responsibility for not only resolving the discordance between variants from different labs, but reviewing all the variants at least every 2 years to make sure there's not new information that's emerged from somewhere to enable a more enhanced interpretation of a particular variant or a change in the interpretation of a variant. So, in conclusion, this whole process, systematic approach to variant pathogenicity classification, is supported by multidisciplinary committees and provides the most reliable assignment of pathogenicity of variants, which is published with a 3-star rating on the ClinVar database when it's been done. This leads to the classification of that variant that's been through this process to FDA approval. Of course, that's a pretty major driving influence in terms of the world's interest in this process. Is that if there's an FDA approval, it means a lot around the world. And this is what you have to do to get FDA approval of the interpretation of a particular variant. The Insight Committee brings intercontinental expertise to the process, building on the experience of their older variant interpretation committees. Algorithmic derivations of the criteria can facilitate classification process, particularly in assigning variants occurring frequently in population database and missense variants to being benign. Biocurator experience and training is essential to the building and growth of the experience, both internationally and locally. Insight holds critical expertise relating to the GI cancer predisposing genes and integrated with the understanding of the ACD criteria. And the Insight ClinGen VCP welcomes anybody from around this room to contribute to their relevant expert participation. That's it, actually. Thank you very much for the opportunity.
much. Great talk and great work. I just want a quick question. You mentioned the thresholds for the allele frequencies. Is that the first step in the algorithm? So is it anything that's detectable, or there's a threshold for each of potentially benign or pathogenic in terms of your allele frequencies?
Yeah, that's a good question. We'd like to think it's the same threshold, but it's actually different. The threshold for determining the possibility of pathogenic is more rigorous than the threshold for assigning benign nature. And that exact point was one of the few points that was queried by the sequence variant interpretation committee, the overall ClinGen process. When we submitted our work, they noted that we had differential thresholds for benign versus pathogenic, and they queried that as to why that should be. But our committee felt it was much more important to be rigorous with assigning to accepting something as pathogenic or possibly pathogenic on the grounds of allelic frequency than dismissing it as benign. So I think that does vary between what happens in one variant curation expert panel to another, but ours felt that we should have a differential frequency. And of course, that frequency varies from gene and disease to gene and disease because of the prevalence of the disease in the populations varies gene by gene.
Thank you.
Thanks, Dr. Macrae. I really enjoyed the talk. One of the interests to me is the BMPROA gene and some of its functions. And I find it really interesting that you're about to publish, and I'm excited to read about some of these more functional annotations. If I had a follow-up and found out it was juvenile polyposis, a Hammer-Normal, and it was very mesenchymal in features, I'd be really excited to know that it was just a BMPROA, maybe loss of function mutation, and there was no SMAD4, there was no activating oncogene. And I wonder if you think about some of the more additive things going back to Ian Kinsler for Global Student 98, talking about the conversion rate of a JPS polyp being so low without an epithelial oncogenic activation signal for a sort of functional diagnosis with other oncogenes. Whereas if you have an APC Wnt pathway driven polyp that is highly epithelial, your progression, your metastasis risks are much greater. Can you talk about maybe some of the things outside of just looking at functional variants and some of the combinational mutational spectrums?
Yeah, I think I can address that by work that we're doing, not in the APC gene, but in MutYH and NTHL1. I have another student who's assembling the information on the cosmic signatures in the cancers that arrive on the different inherited background. So there are specific combinations of mutations that occur, represented by cosmic signatures, that can be associated closely with the DNA background in the germline. And that information can potentially be very useful in terms of pushing a variant of uncertain significance in the germline into a pathogenic category, because you're seeing the cosmic signature in a cancer in a patient that is typical of, say, MutYH germline mutation. So you can say, therefore, that the germline mutation is probably pathogenic because it's producing that cosmic signature in the tumor. I think that's a little way to the way you're asking the question, but I guess you're really expanding a bit further if we apply that more broadly and note the mutations assembling in a particular polyp. We might be able to expand that into something similar. Probably cosmic signatures are the way to go because it represents some understanding of the range of variants that occur in particular circumstances.
Just a follow-up to that is, and I'm just guessing that you're starting to gather is some of the data can become more longitudinal where patients are going to get repeat sequencing. I suspect if they haven't already, And I'm wondering if your database integration is starting to see when new mutations progress and occur from different platforms as we see more cancer centers across the world are starting to do deeper, bigger, more integrated longitudinal sequencing. Are you going to start to be able to put longitudinal, map patients as they progress?
Yeah, I think we will, but even before that, we can look at synchronous polyps to see how consistent the signatures are between two polyps occurring in the same individual. To see how consistent the signature is, even at the one point in time with synchronous polyps, or cancers for that matter. Longitudinally, I think there is information evolving, and Swati might remember this from our insight meeting, but I was particularly intrigued by the German work that is identifying the sequence in which mismatch repair deficiency occurs in polyps, stratified by gene. So with the MLH1 gene, the initial event is microsatellite instability. Even at the isolated crypt before there's any polypoid evidence, you've got a range, a whole host of mismatch repair deficient crypts, just isolated crypts that are mismatch repair deficient. So that is the initiating event in MLH1. Whereas with MSH2, it's the reverse. There's an APC mutation that happens first, and later on, mismatch repair deficiency comes in and accelerates the process. So that's in a way, that's derived from cross-sectional information that's extrapolated to what might be happening longitudinally. I think that's the best I can answer in terms of the current information.
Any other questions?
So I'd just like to say that you recognise from this that this is a global problem. This is not just, in my experience with DNA biocurators, is even, well, particularly in the environment, in our environment, they're all siloed, they're colloquial. They don't look much beyond what they can see outside their own institution. Whereas this is a global problem that requires global addressing, sharing, and understanding DNA variation from a global perspective. So don't be afraid to share.